to all joining us today. Today's talk's title is Foot and Mouth Disease, the Practical Consideration. My name is Adele Krach, and I'm from Food Focus, and I want to thank the Dairy Standard Agency for sponsoring this webinar. The Dairy Standard Agency acts as the independent organisation of the organised dairy industry with its members MPO and Sampro. The main purpose of DSA is to promote the improvement of the quality of milk and other dairy products in the interest of the consumer and the industry. One of the 17 projects of the DSA is information and education, which deals with webinars covering the entire dairy value chain. And this is yet another webinar doing exactly that. It's a pleasure to hand you over to Jompie Burger from the DSA. He will introduce the speakers and the topic, and he will also be handling the questions after the presentation. Welcome, Jompie.
Thank you. And technology is not always the best that we expect it to be, but it's still wonderful to talk to basically from the north and the south of our dearest planet. Yeah, today I think the discussion that we are going to have is very contentious. It's something that we all need to know about right throughout the value chain, and it's very important to pay attention because we basically divide the detail of what we're going to hear today into three sections. The one is from Dr. Francois to hear more about the introduction of foot and mouth disease, listen to all the causative organism, the prevalence, the mortality, and the finer detail of that, but also what can be done. So from Dr. Ben's side, we can also hear in terms of the importance of proactive measures, and I think this you will also hear from Dr. Francois, that's correct, to be taken by industry members in terms of risk mitigation. You can clearly hear from these two bullets and the third one, the actions and responsibilities by industry if there are no suspect or suspicious activities in your geographical area, and even if there's an outbreak. So very important, what is this disease? What is our role as an industry at primary and secondary industry level, but also what is the activities when the outbreak occurs, but also elements of what is the state's responsibility to this effect. I'm sure that we're going to have some questions today and lots of questions, but first of all, I want to hand over to Dr. Francois. Dr. Francois is very well known in the industry for all his activities and the role that he plays in terms of disease.
I really appreciate this opportunity that I can talk to all of you today. So to just give you a short introduction of myself, I worked with FMD for nearly 20 years, and I've done this work at the Agricultural Research Council of South Africa, and specifically the site at Onderstepoort. And I worked with colleagues from all over the world, because FMD is a worldwide problem and not just a local problem. So, and today I want to tell you a little bit from what we learned and what I know from the literature about the transmission and pathology of foot and mouth disease virus. And this information will be important for you to understand the control measures and also to understand what you need to do at farm level to prevent the disease or to get it on your farm. So to give an introduction, okay, can everyone see my screen?
Yes, thank you, Francois. It's perfect.
Okay, I'm just trying to forward. Oh, there it goes now. So the important thing to remember here is foot and mouth disease is caused by a viral infection, and this viral infection is very specific to all your cloven-hoofed type of animals. And since many of the cloven-hoofed animals play a huge role in our farm animals, these include cattle, swine, goats, sheep, and then also in certain regions in the world, the water buffalo. It's a viral disease. There's 7 distinct types of this virus that is circulating, and this virus circulating especially in wildlife. Now, the important thing is, although there are 7 distinct serotypes, which implies if an animal has been infected with one, it may not have antibodies that will protect against the other one. So there's no cross protection. But clinically, they look the same. So how in the world we define the status of FMD in different regions. And from this map, the gray areas that you can see here is all the areas that have FMD. The green areas is without FMD. And as you can see, in 2017, South Africa has been free of FMD. This situation changed very quickly. And actually, we lost our FMD-free status by 2019 due to outbreaks that happen beyond the control regions. So this is what we look at in terms of South Africa FMD status in 2022. And this is a result due to the outbreaks that we experience in KwaZulu-Natal, as well as the other provinces. So in order to control FMD, so how does the situation change from the green areas that I showed in the previous map to the gray areas? It all boils down to the control, effective control. And to do this effectively, we need to understand the behavior of this virus, both in domestic animals as well as in wildlife. Subclinical infections, persistent infections, carrier state, etc. So, and it's on these topics that I want to spend some time today. So, and the purpose of my presentation is to give you information, or at least give a reference if you need to go and look up something that you're not sure of, because there's a lot of misconceptions out there. Now, foot-and-mouth disease are found endemically in wildlife in Africa. And the wildlife animal that are especially important here is the African buffalo. African buffalo are persistently infected with foot-and-mouth disease. And for example, in the Kruger National Park, we have found that by the age of 2 years, young buffalo have been exposed to the 3 SAT serotypes that are found here in Southern Africa. High proportion of outbreaks are caused by one of these serotypes, SAT2, which every now and then jump over from buffalo to cattle, and then there's an epidemiological pattern that form in cattle. Mild and subclinical infections is really critical here, and is the reason why many times foot-and-mouth disease are missed, which implies that foot-and-mouth disease can go on for quite a while without being seen. So we'll spend some time on that also. Let me just stand still on this topic of the epidemiological cycles. So I've mentioned now in my previous slide that buffalo plays a role. The disease is endemic in buffalo. So we know we have these endemic cycles in buffalo, and there are basically 2 mechanisms how the virus are maintained within buffalo, and the 2 ways is by means of acute infections, and some animals in that herd then become persistently infected and can carry that virus for 2, 3, up to 5 years, and is then a source of new virus in the new calf season, for example, where the calves become born, and after 6 months they lost their maternal antibodies and become susceptible. Every now and then the foot-and-mouth disease escapes from buffalo and can infect cattle. This is a relatively rare event, but since foot-and-mouth disease is so infectious, it can start an endemic cycle in cattle, and that is what one would like to prevent. Now, what is the situation now in South Africa? We see outbreaks. We have outbreaks in Northwest Province, KwaZulu-Natal, Free State, there's outbreaks in Limpopo on an annual basis. Can we talk about a endemicity? I think, and others share my opinion here, is that there is most likely an endemic situation developing in certain regions, and I'm talking more about the northern parts of South Africa. So the current outbreak that we experience in South Africa is not endemic yet. So it can be stopped, and if we are successful to stop this, this infection will become a dead-end infection. Now, as I said, the northern part of the country, there are always things continuing going on in terms of FMD outbreaks. And some of these outbreaks, we see the same virus on a genetic basis. We can identify the virus in cattle in different years. So, which implies there is some sort of endemic situation happening there. And for this region, the control in South Africa was always focused on having an area around the Kruger National Park, which contained buffalo, to vaccinate cattle there and to create this immune barrier in cattle, but also have physical barrier between animals that are persistently infected, like the buffalo, and susceptible animals, which is your domestic animals. And it was successful for many years, so we do know that it worked. How does FMD work in cattle? So the moment cattle get exposed to the virus and the virus get entry to the animal, there's a first round of replication and a primary viremia that develop. So following exposure and replication, there's primary viremia, and from that site of primary replication of the virus starts to spread through the blood and through the system and can bind to epithelial cell and other cells that are susceptible to this virus and start replication there. So that's why typically one see infection in the mouth, tongue, dental pads first, and later on in the infection, it spread to other areas of the animal, teeth and hooves. So, but there's a period where virus replication takes place, virus shedding takes place, but one do not see clinical signs. By the time you see clinical signs, there have been a lot of virus shedding taking place. And then in the last part of this figure, in some animals, they become persistently infected. And I will talk about persistent infections in a moment. So just taking that figure into consideration, by the time an animal has been exposed and virus replication has taken place and the first or primary viremia has taken place, until the time that clinical signs have been started to show in the animal, there's a lapse of 1 to 3 or 4 days. And that period where the animal is subclinically infected is actually a very dangerous period. And that's where a lot of virus has the potential to escape from that animal and infect other animals. So what is the route of infection? And how does foot and mouth disease virus get entry into the animal? Of course, direct contact of susceptible and infected animal is especially in our area, one of the most important things. And anything that infected animal touch, whether the saliva ends up in the food, in the water troughs, or any mechanical means, apparatus that are used to handle that animal, all of those are possible ways to transmit the virus. In some areas in the world, airborne transmissions is quite important. And yes, it is a respiratory virus. It infects in the upper respiratory system, the nasopharynx, and then also in the lungs as a secondary site. However, with our conditions in South Africa, I'm not completely convinced that airborne is one of the major transmission routes for foot-and-mouth disease. Sexual transmission can also take place, and I think there's 3 factors to consider whether a susceptible animal will get infected. That's the quantity of the virus that are released in the environment, the ability of the virus to survive that environment. So we will look into the stability of this virus in a moment, and the quantities that are required for infection. Here is another summary of that. So the entry routes is really important. I took this slide from a manuscript, and it's just to highlight, and it's not, the previous slide is not just my words, it is scientifically determined what the entry routes are for this virus. What is the minimum dose during this route of infections? So we looked at 4 things, or this paper looked at 4 things here. Inhalation of the virus, intradermal infections, and then also if one injected intramuscularly, or then nasal instillation. So important here that cattle are very susceptible in inhaling this virus. Now, if you take all of this information together with the behavior of cattle are very social animals, and they need contact with each other. They lick each other, and so you can just imagine how quickly and how rapidly this virus spread by any of this means. So pigs require 80 times more virus to get infected via inhalation, but they're more susceptible in other ways. For example, the direct contact, and also their hooves are very susceptible to get infected via direct contact. Once the virus enters the animal, the cells in the mucosa, linings of the mouth, the tongue, the lymphoid tissues in the mouth and area and pharynx area, these are all cells that are very susceptible to the virus. The moment the virus touches that cell, it will enter and it will release its RNA and replicate. So it's important to know that the virus can be detected in the oropharynx 1 to 3 days before the onset of viremia, and this is this subclinical stage that I keep talking about. Now, the virus can not only be detected, it's high amount of virus that can be detected, plus the animal is infectious at that point, and that is before one see any clinical signs. The viremic stage and takes about 3 to 5 days. Around 7 to 14 days, all the lesions in the mouth disappear and you can't see anything. The animal has started to recover. Secondary phase of replication. So I kept talking about the primary viremia. Now the secondary viremia is the coronary band in the hooves, the tongue and tendon pad, teats, and then also the heart, especially in young animals. And some young animals actually die due to infection of the heart muscle. So it does happen, but generally the mortality rate of foot-and-mouth disease infection is very low. Some farmers out there ask us, you know, we don't see any animals die. Why do we have to vaccinate? Why is it such a big issue? And it's difficult questions to answer, because yes, not a lot of animals die, and I think in this outbreak currently, I didn't know of any animals that died due to foot-and-mouth disease virus infection. It does happen in other areas and with certain serotypes. So virus can be excreted 1 to 4 days, 1 to 3 days before the onset of clinical signs. I keep saying it because it is important, and it needs to be considered when you trying to move animals, and when you're trying to actually protect your farm, not getting these infections on your farm. So virus excretion usually ceases around 4 or 5 days after the appearance of vesicles. So I would say from the time that the animal has been exposed, 7 to 14 days later, the animal has recovered completely, and no virus will be excreted further. That is in most of the animals, not all of them. But a lot of virus is present in those epithelial that come up and erupted vesicles. And remember, once it jumps into pigs on your farm, pigs have the highest excretion levels. And taking now this into consideration, pigs excrete high amount of virus. Cattle need very little virus to get infected. I added a few additional slides because I thought, since I talked to you guys that are in the milk industry, it is important to know. We know that viruses are shed in the milk also, and we know this because FMD outbreaks have been linked to contaminated milk. So FMD virus can also be shed in the milk before we see any clinical signs, coming back to that subclinical stage of infection. And remember, it's a huge amount of virus, so those animals are infectious. And the virus can survive for quite a long time in raw milk. 6 days if that milk is stored at 18 degrees Celsius, and up to 15 days at 4 degrees Celsius. Now, these experiments have been done with some of these other serotypes and not with our serotypes that we get here in South Africa. They are a little bit more sensitive, but still you can rely on a good 7 to 14 days survival when the milk is stored at 4 degrees. Here is information also from the literature to give a little bit of an idea of how much virus are present and for how long it is present in different samples. When we talk about saliva sample, nasal secretions, or milk samples, and as you can see in the milk samples, especially in cattle, the virus concentration or amount of virus is almost 10 times higher and are present for a much longer time than in the saliva. The stability of the virus in different environments. The virus is sensitive to environmental changes. pH, that is acidic versus alkaline. The virus best functions around a normal pH 7 or physiological pH. Temperature is very sensitive to temperature. Temperatures above 37 inactivate the virus relatively quickly. pH, when the pH drop to acidic levels of below 6, the virus will fall apart relatively easy. But what does it mean? How does the virus survive in different things? In hay and straw, for example, people have observed 20 weeks survival of foot-and-mouth disease virus in hay and straw, 14 days in dry fecal matter, especially in the summer. In fecal slurry in winter, up to 6 months survival. 3 days survival in soil during summer days, 28 survival in soil during winter days. So if you want to know when and how to inactivate virus after you had infected animals in a certain area on your farm, keep this in consideration. So remember I said most animals clear the virus from all their secretions within about 14 days. However, there is a small proportion of animals, cattle, sheep, goats, that become persistently infected. And that persistent infection we define as there is still virus replication, and we can detect this virus replication 21 days or longer after the primary infection. So After the clinical signs disappeared, some of them can have replication of the virus, and these animals can also transmit this virus to susceptible animals. It is real persistent animals because there's virus replication and they're infectious. And with time, as time goes by, this infectious virus disappear, and about five months and sometimes up to 3 years, it will disappear completely. But the animals can become persistent for quite a while. I'm not going to say too much about immunity. It's more science related. But if you have more questions, you can ask me. Calves can get antibodies from their mothers. Also in the colostrum, there's antibodies. And by around 6 to 7, 8 months, the maternal antibodies disappear completely, and that calf will be susceptible. Antibody responses are important to neutralize the virus and to remove the virus from the animal, but any antibody response is always dependent on a T lymphocyte response also, and we do know that the virus replicate in some of the lymph nodes. So I'm just going to spend this 1 minute on vaccines and vaccine efficacies. We're going to enter most likely a huge vaccination campaign in South Africa. I hope and I believe, and I'm sure Dr. Ben will tell us a little bit more. We need to remember there are inherent limitations to the vaccines, and the duration of antibodies are relatively short with the current vaccines. By around 3 to 4 months, antibodies levels start to disappear and animals become susceptible again. Keep that in mind when we do a vaccination approach. If primary vaccinations doesn't solve the problem, there is most likely a need for a second round of vaccination. So one thing I talked about, persistent infection in some animals. So under natural conditions, if an infection follow its natural way of going through the animal, around 10 to 15% of animals become persistently infected. However, when one vaccinate animals, yes, it prevent clinical disease from forming. And remember, vaccination do not stop virus replication. It just prevent clinical disease. However, there are a larger percentage of these animals that have been vaccinated and exposed to an infection that can become persistent infections. And people say that up to 50% of those animals can become persistent. I'm not going to say too much about this. This is information and studies that I've been involved with for many years, and that is the interaction of the virus and the interaction between buffalo and cattle. We do know that carrier buffalo do carry the virus and can shed the virus at certain points in times in their lifetime. And when they come across susceptible animals, they can infect susceptible animals. These things have been observed for on and off in terms of experimental infections, but also in the field. And Dr. Ben can confirm these type of observations that have been happening in the past. Carrier or persistently infected buffalo can transmit a virus to susceptible cattle. What that trigger is and that whole mechanism, we're not 100% sure. But once the virus gets into the cattle and cattle to cattle transmission takes place, then that becomes a serious critical situation where endemicity in cattle can take place. So one of the things that I believe we need to do more also in this outbreak is surveillance type of work, and actually bringing in the ProBang as a sampling tool and start analyzing samples taken by ProBank. And this is because we miss clinical signs in the subclinical infection. We miss clinical signs during the persistent infection. But those animals can still be infectious. And if they get in contact with susceptible animals, it's a problem. So how to conclude? FMD is highly contagious. There's unique epidemiology in Africa. And once it is established in livestock, an endemic cycle can start to develop. Subclinical infections, persistently infectious animals are real and are the real risk here. Longevity of antibody responses following vaccination is sometimes quite short, and keep that in mind in control. I think this is what I have to say. I think I'm 3 minutes over. Sorry for that, and thank you for this opportunity.
Thank you, Adele, and also thank you, Dr. Francois. There's years and years in research of being done in this field, as you can hear from Dr. Francois, and we highly appreciate your involvement in this work together with all the other role players. I think we still have a lot to learn, but I think one of the most important things is what we as an industry also must contribute to the prevention of this disease. And at this note, I would like to thank you and also ask Dr. Ben to take over. Dr. Ben is also very much involved in this process from a regulatory environment, and I want to thank him also for participating. Dr. Ben is the current Deputy Director Animal Health and Foot-and-Mouth Disease Coordinator of the Department of Agriculture in Mpumalanga, and also the other expertise that we thought is the most appropriate person to talk on this subject. So Dr. Ben, if you can take over, we're quite keen to hear what you have to say.
I will talk about the status of the disease in our country and then about the control. And I'm glad that Mr. Berger said that the dairy industry can contribute. So, like the state can also contribute, because nobody alone can tackle and control and prevent this disease. We need it to be a joint venture. So thank you for the opportunity. Firstly, I want to show these two photographs which I took just to show the variance in the intensity or the severity of the lesions that we can see. On the right-hand side, that was the biggest lesion that we had in an outbreak of affecting 12,000 cattle. Easy to miss. The left-hand one is more conspicuous. That was in a few hundred cattle, but that's just the variation, and therefore it's important to be on the lookout and not to disregard some small lesion as not being foot-and-mouth disease. I'll say some things on the status of the disease, the different zones and areas in our country in the past and at present, and then a short few remarks on early detection and reporting, which is very important, and then just how, in general terms, an outbreak is controlled. I'm not including prevention into this talk due to lack of time, but that is a routine thing. It's not now in the spur of the moment necessary, I would think. The status, as you can see in the top map of the world, that's just the distribution of the different virus serotypes. And then the bottom one you've seen earlier with the current outbreaks in Kazakhstan and in Indonesia. And our country is not green anymore, as Francois said. Now, according to the World Organization for Animal Health, at the moment we are regarded as an infected country, even though the infection is just in certain parts, but that is how the recognition is operating. We've got veterinary legislation, the Animal Diseases Act and the Animal Diseases Regulations, which is currently applicable. And everything that the state does in connection with disease control must emanate from this legislation. We just can't go and tell anybody to do anything if it's not backed up by legislation. Foot-and-mouth disease is a controlled disease according to our legislation, and the whole of the Republic of South Africa is a foot-and-mouth disease controlled area. Now, in the regulations, the control measures are very generic and very short, not much longer than on the slide. With regard to susceptible animals, they may be vaccinated as determined by the director, and contact between buffalo and cattle is illegal. Then contact animals, that's on animals that were in contact with infected animals, they must be isolated and further dealt with according to the determination of the Director of Animal Health. Infected animals must be isolated, and they may be vaccinated or otherwise disposed of or dealt with as determined by the director. So you can see there's nothing specific there, and the reason for that is that outbreaks can vary tremendously between small holdings, big farms, big feedlots, dairies, game reserves, etc., communal areas, commercial farming areas, and therefore there's nothing specific. That is more specific in other documents and procedures and manuals and procedural notices, etc. Now, the routine control areas in our country consist of the infected zone where there are infected buffalo. That's basically the Kruger Park and adjacent farms and other reserves, as well as the Ndumu and Tembe Reserve in the north of KwaZulu-Natal. And then around that, there's a protection zone, which consists of one part where cattle are vaccinated every 4 months, more or less 10 kilometers wide, but it depends on the geography and the animal populations. Then another zone of about 10 kilometers, which is also part of the protection zone, but where there's no vaccination. That is a very important zone for surveillance because those cattle are naive or susceptible to the disease. Then there's the rest of the country is the free zone, which consists of a small part in KwaZulu-Natal, which is a high surveillance area where there's movement control. And then the rest of our boundaries, our inland boundaries are forming the high surveillance area. The rest of the country is the free zone. Now, apart, or in the past, we had a free zone without vaccination that was recognized internationally. We lost it 3 times. The last time was in 2 January 2019 because of the Limpopo outbreak. As you can see there, the outbreak moved to the west of the yellow line. That's into the free zone in January 2019, and then we lost our status for the 3rd time. So hopefully in future we will be able to regain it again, because that is favorable for our country for exports of various animal products and live animals. Apart from the permanent or the routine zones, excuse me, we've got at the moment 2 disease management areas, one in Limpopo, one in KwaZulu-Natal, as you can see on the map. Those diagonally striped areas, which was last updated in May, and then also various pieces of land that's quarantined at the moment due to outbreaks. Those are all those red spots in Mpumalanga, one in Gauteng, one in Northwestern Number, in the Free State Number, and then also in KwaZulu-Natal, some outside of the disease management area. Now, what is very important is that because foot and mouth disease is a controlled disease, and because it's highly contagious, can spread easily, and cannot controlled by any individual person or farm or institution, the state has the obligation of coordinating the control of an outbreak, but the state cannot do anything if it's not early detected and reported. And therefore, any farmer, farm worker, veterinarian, person working with animals in abattoirs, wherever, if any foot or mouth lesion is seen, and it's easily remembered, just go back please, easily remembered from the name of the disease, that must be the first suspicion or the first differential diagnosis to be thought of, and the first one to be either ruled out or confirmed, before other things are thought of, like blue tongue, lumpy skin disease, injuries, whatever, because of the major consequences that it can have if it is the disease, and especially if it's not reported. Because if reporting is delayed, then the spread can be enhanced and losses can increase. And that happened recently in a number of the outbreaks that we have at the moment, where reporting was very late. In the meantime, there were some movements of animals, some auctions, and so on, spreading it to other farms and affecting other businesses and farmers.
Sorry,
Now,
Dr. Ben.
according to our legislation, any person in the country, especially veterinarians, laboratory staff, but that can you hear me?
Yes, Dr. Ben, sorry, there was a slight delay. Can you please just start with the slide again?
Okay, thank you. According to our legislation, any person in South Africa is obliged to report immediately, verbally, followed up by a written report, any suspicion or a confirmation of any control disease, including foot and mouth disease, to the Director of Animal Health, to any state veterinarian or any animal health technician. That's also a suspicion, even though it may not be typical. That is on mouth lesions, on feet lesions, as well as on teeth lesions, so that it can be further investigated. Thank you. Now we come to the control of an outbreak, and I just want to repeat that control is a joint operation between the state, the industry, owners, and all other role players in the particular industry. It starts with a suspicion that's noted by somebody, then early reporting, then communication to all the role players, also to the neighboring farms. Then the first thing that is done then is quarantine of the area, even though the disease may not have been confirmed. That is to immediately prevent movements and to prevent spread. Now, quarantine doesn't necessarily mean no movements. It means no movement without a permit, because it may still be the case that during the course of quarantine that's imposed on animals, that some products or some animals may move. But that's then on permit, so that the risk can be minimized of that movement of spreading the disease. Investigation is done with the purpose of confirming the disease or ruling out the disease, and then once it's confirmed, also to try and find out the extent of the disease, which animals are on the farm or on the premises or at the dip tank or in the feedlot or in the dairy, and which animals are affected. Once it's diagnosed, and it's diagnosed by laboratory testing, it may be only on blood tests, but preferably on a tissue sample from the lesions from the mouth, so that the virus can be isolated and identified and further investigated. Once the diagnosis is done, then some outbreak areas are determined. Now, this is just in general, because it may differ vastly according to the circumstances of the outbreak. There are just two examples on those maps. The one is in a feedlot where there's obviously an infected area. That's where the infected animals are, or the infected herd. And once it's diagnosed, the whole herd or all the animals that's in contact with that confirmed animal is regarded as infected. Then an infected area is determined. If possible, around that, the animal-free area. The bottom map, that's not possible because it's a communal grazing area where there's no fences. It's not possible there to create animal-free area. But if possible, it's of great assistance in curbing spread of the disease. Then around that, the quarantine area where there's no movement allowed initially, at least of any animals or animal products. And then a surveillance area around that where only inspections and other surveillance is done in order to detect any spread. Initially, the quarantine area may be, and is supposed to be, quite big, which may be made smaller later on as results are obtained. Then there can also be a vaccination area if vaccination is an option in the control of the particular outbreak. That's not an area, a specific separate area on its own. It may entail several of the other areas according to the risk analysis. Next one, please. Then once the areas have been determined, then movement control, actually all of these things happen simultaneously. It's difficult to put them exactly in a chronological order, but movement control is then imposed on animals, obviously, on products of animals. Can also be other products like, for instance, feed or hay or dung or manure from animals on people working there, access control, and then also on vehicles, because all of these commodities can contribute to the spread of the disease. If possible, roadblocks are put up. It's becoming more and more difficult and not so very efficient to put up roadblocks. And in this regard, the cooperation of the community and of the farming community and the industry is very important, because it's not only the state that can now prevent these dangerous movements from taking place. Tracing is done backward from the form of infection and forward to try and find out where it came from. Not that it influenced the control on the farm, but in an attempt to also then detect other places of infection, so that control can also be done there. And in this way, many of the farms that's currently infected was or were detected. The tracing does not only entail animals, but also products that moved, people and vehicles, even though animals is the main method of spread of the disease, but it is also important to do tracing on other ways of spread of infection. Again, for the sake of early detection on other farms. Then the issue of vaccination. Vaccination is not done in all outbreaks. It must be done with a specific purpose, and the one is if it is sort of the only method that can be done where movement control is difficult, where there are no fences, where there is no proper individual control over herds because of the grazing or the grazing setup. Then vaccination is done to try and spread to prevent the spread of the disease, and also in cases where there's a high density of animals, like for instance in a feedlot, where if in the absence of vaccination, the viral load that's produced by excretion can be so big that it can contribute to the spread of the disease, especially if there are other animals close by which cannot be moved. So that is not in all instances indicated. In a small herd that's quite isolated, or even in a bigger herd that's isolated, herds that are kept extensively and which can be separated from other herds by an animal-free zone, their vaccination may not be indicated because it has its disadvantages. For instance, it makes the detection of the disease more difficult, the clinical detection, because it suppresses the clinical signs. It does not prevent infection, as was said by Francois also, but it does suppress the clinical signs and the amount of virus that is excreted. In the case of vaccination, the fate of the animals is not necessarily to be slaughtered or to be culled afterwards. We can also do vaccination to live, but in cases where animals were in any case intended to be slaughtered, like for instance in a feedlot, then obviously they will be slaughtered at some stage, even though it may be then delayed slaughter. In the case of vaccination, sorry, it's important to mark the animals so that they can be identified, so that we know where vaccinated cattle are in the country, for the reason that we had a free zone without vaccination. And if our purpose is again to strive for recognition of a free zone without vaccination again, it's important to know where these cattle are so that they can be monitored until they are not having antibodies anymore. Vaccination can be done once, if that is enough to suppress the disease. In certain areas, especially in communal areas, we vaccinate up to 4 rounds to control the disease, because the spread is so much easier where there are no fences or animal-free zones that can be created. Now, the 3 control options of aphtization, destruction, compensation, I'm not going to mention, because at present they are not applicable in our country in the situation that we have. Then surveillance is done not only on the farm where the infection is. Once it's diagnosed and confirmed, it's not really of practical view to do a lot of surveillance in that herd itself, but it's much more important to make sure that all the herds around which might have been in contact with the infected herd, as well as herds on farms where there might have been a connection to by animals that moved just prior to the confirmation, or vehicles that moved, or people that moved, or products that moved. Surveillance is done amongst those animals to make sure that they have not been infected. In many instances, we must first do a quick census to find out exactly what species and what the numbers are of the animals in these control areas, so that we can make sure that all the animals are covered by surveillance, which include clinical inspection, mouth examination, also feed examination, but it's more time consuming and not so easy for practical reasons. Then samples are taken, blood samples, to detect antibodies, which is not the virus, but just the reaction on the virus, which may be the result of infection, but it may also be the result of vaccination. Virological samples taken, that's tissue samples, hopefully containing virus if there are, to try and isolate the virus. And then lastly, also to type or sequence that virus so that we can form an idea of where it's coming from by comparing it to viruses isolated earlier, wherever in the country. And in that way, we can also determine often the cause of the infection, which can then be used in future to prevent the similar event. What is of importance is that the clinical endpoint must be determined. That is basically, for all practical purposes, the date of the last case of the disease, because that will determine now the exit strategy out of this outbreak, which may be removal of the animals or retention of all the animals. But eventually, the end goal is to lift quarantine so that normal farming operations can continue. If controlled slaughter is the option, which is, like I said earlier, the case where animals would have been slaughtered in any case, and that is not compulsory, that's a choice of the owner, then it depends when the slaughter is taking place, what the losses will be. And without going into detail, there are certain things that must be done during controlled slaughter which may cause losses. Carcass maturation must be done in all cases. That's in any case done. That is to lower the pH of the meat, which is killing the virus. Then if slaughter is happening quite soon after the clinical endpoint, then destruction of the head, feet and offal must take place, but it can also be processed by methods killing the virus. Then carcasses must be deboned and deglanded up to a certain stage, and if they are slaughtered within the first 14 days, the meat must also be processed, in that the meat must be cooked so that virus can be destroyed. Then if depopulation is taken place, that's either by controlled slaughter. It can also be by removing animals to another farm which is infected already, or for whatever reason, but that is all done while the farm is still under quarantine, so that will be done in a way that it will not cause another outbreak. If animals are retained, then quarantine cancellation is determined by a certain period after the clinical endpoint, and then also by serological tests to make sure that there's no more virus activity. Obviously, if there are any clinical signs in that period, then the whole process starts all over again. If some of the animals to be retained have been vaccinated, it's the same process, that they must be tested until they haven't got antibodies anymore. Infected animals will have antibodies also, but that will also wane after a while. If animals are vaccinated only once, then the antibodies will not last very long, so it's not years and years and years that animals may not move or remain under quarantine. If possible, after removing of animals, cleaning and disinfection can be done. That is more in intensive setups like dairies or in feedlots. But in field situation, that is not impossible. Impossible. The repopulation can be done at a certain stage, depending all on the setup and on the size of the farm and where animals can be brought into. They will be subject to serological testing, to blood tests, to make sure that they haven't picked up the disease from somewhere on the farm. And then if everything is in order and there's no evidence of virus presence or virus activity or transmission anymore, then quarantine can be cancelled. And what is then of utmost important is the continued biosecurity of making sure that this thing does not happen again. And there are basic guidelines for that. I will just in very briefly mention that what can work, which is already legislated in some instances, is that to make sure that before animals are moved, that they've been on that premises for 28 days, that no other animals have been introduced, and that they showed no clinical signs in that period. Now, that cannot be easily certified by a veterinarian, but it can be declared by the owner, because I think especially in the dairy industry, animals are seen very often, and owners and farmers must, and managers and actually workers must be acquainted with signs, not only of foot and mouth, but of other diseases also. So it's important that before they move, that no such signs have been observed, and that there were no abnormal mortalities. And then they can move whatever legal requirements there are in the area at the time. It may be in a controlled area, it may be wherever there's permits required. And then on arrival, if at all possible, because it's advisable at this stage, that cattle are put in isolation or in quarantine, separate from the main herd or herds on the farm, kept there for another 21 days, preferably as part of the farming system already as far as nutrition and management is concerned, and then health checked after that by a veterinarian, preferably, before they are introduced onto the farm. If everybody is doing that, the virus will have a very hard time of spreading. And if we can aim at that and promote that and implement that, then our future is bright and our export markets may be opened again in future. I thank you.
Good. I'm back. Thank you, Adel and Dr. Ben. Thank you so much for sharing this information with us. As I can see from the platform of listeners, that all the people that registered, we come from different backgrounds and from various roles in the dairy sector. But thank you for enlightening us on this. We have some questions indeed, and on the panel we have Dr. Ben, and we have Dr. Francois, and then also Dr. Mark Chimes. But one of the questions that came through is in respect of if a herd is quarantined, what happens to the milk? Is it permissible then for the transportation of this milk to a facility where it can be processed to ensure that it's safe for human consumption, Dr. Ben?
Thank you. Yeah, I said that quarantine doesn't mean no movement, but it means controlled or safe movements. So without giving now a general answer that it can happen, the situation will be looked at. Most or usually milk's pasteurized in any case, which is quite effective against foot and mouth disease virus. There are international guidelines for the disinfection of foot and mouth disease, which we will then follow in this instance, especially if milk is used for the feeding of other animals. Then double pasteurization or ultra high temperature treatment will be necessary. But then we will also look at the transport. If it's on the farm, it's of course more safe. If it's at some other processing plant somewhere, then we will just look at the safety of the transport and of the vehicles. But obviously, we always try to strike a balance between what is safe, but what is also advantageous for the continuation of the farming operation. Because being only scientific will say no, milk may improve.
I assume then, Dr. Ben,
sorry.
Sorry. Now, you finish first. There's a next question.
I just want to say that according to the circumstances, it can be looked at, but it is not impossible for that to be allowed.
Okay. Any process or any steps in this direction would be subject to the approval of the provincial state veterinarian, I assume.
Yes, while a herd is under quarantine, which include all products and feed and so on, then all movements must be discussed and risk assessment will be done. And according to that, there will be permission to move products or not. Because if it is endangering other farmers and other herds, then obviously it's not the right thing to do. Thank you.
The next question that we received was regarding the vaccine rollout that was alluded to earlier. If a commercial dairy herd is vaccinated, either by choice or necessity, how would this impact the dairy processor's ZA certification for export of dairy products? Dr. Ben, I suppose this one is for you as well.
Well, that will depend on the requirements of the importing country. But usually that milk will not be exportable because it can have antibodies in the milk. If there's a possibility of still virus in the milk, then it will not be allowed. So that will be a limitation.
There's a follow-up question in this regard. Is there a test that can be done on raw milk to detect the presence of foot and mouth disease?
You'll ask Francois to answer this one.
Thank you, Francois.
Yes, definitely. There are tests that we can do to detect foot-and-mouth disease virus in milk. It's called the PCR test or polymerase chain reaction. Now, in the case of foot-and-mouth disease virus, which is an RNA virus, there is a reverse transcription process that precedes the PCR, but it is a test that are used internationally and can be applied for milk also.
But then, Dr. Francois, these tests can only be done by institutions that is approved for the purpose of this, because it's a state-controlled situation, a notifiable disease. In other words, it's not a test that can be done without accreditation and approval of the department.
Exactly, yes. No, samples will have to be sent to Onderstepoort, to the Transboundary Animal Diseases Center there. They have the capability or the mandate to perform these PCR tests. So anything foot-and-mouth disease related, at this point, it's not allowed for a private vet or a farmer to do the test for himself on site. It will have to be sent to the biosafety level 3 laboratory at the ARC.
Thank you for that one. The question relates to how can the process of vaccination be excelled or accelerated so that there's a definite attempt to speed up the process. Is it something that only the state must do, or could there be an element of collaboration as well to make sure that all animals are vaccinated as quickly as possible?
I suppose it's for me the question. There's no plan to vaccinate, to do blanket vaccination in our country, because the disadvantages are far more than the advantages. So preventative vaccination will only be continued in the protection zone around the buffalo, and vaccination is elsewhere is limited to specific outbreak situations and not preventative at all, because it will influence our export status, and it will mask or hide the disease that it cannot be detected. It will influence the serological or the blood tests that we do, that makes it much more difficult to distinguish whether we've got now a virus or whether it's because of vaccination. So there's no such plan.
Okay.
There are rumors going around, but I don't know of any such plan, which will in any case not be good.
This question relates to the sale and transportation of bull calves, obviously to abattoirs and all places where, or farms where they can be raised. Because obviously the bull calves should not stay on the farms too long for all economic reasons.
I assume this question is not in case of an outbreak, but at the moment, where there's no outbreak, but in relation to the movement ban. Is that correct?
No, this is specifically in the case of an outbreak. What happens to the bull calves then?
In an outbreak on a farm, no animals may move because it may spread the disease, so the bull calves will have to remain there and be fed and raised until the outbreak is over. Because the priority will be not to spread the disease and to contain and eradicate it as soon as possible without causing some more outbreaks to where the
Dr. Benify, the question has been the question has slightly been rephrased. And what about this matter in, let's say, in the Western Cape then?
Is that's now in relation to
on
the
farms
current
where
ban?
there's no current. Okay.
Yeah, at the moment,
Okay,
it's not permissible. It's not permissible at the moment because of the ban until 8 September. We will see what will replace the ban, whether it's extended or whether it's amended, or what will be the situation after that. But I am of the opinion that it will definitely make provision for more movements that is inherent to the different industries, without compromising the risk of spread at all.
Yeah. Good. Dr. Ben, thank you for this. I saw some more questions, and I think we've just run out of time, and I will just ask Adele also to finish off. But from our side, from the Dairy Standard Agency and Milk SA, and also working together with the Veterinary Association of South Africa, thank you so much for your time and for the absolute fantastic work that you do, and that you are so willing to share that information with us. It's truly much appreciated. The questions that's been asked and not answered, we will surely get back to you on that, and we will obviously respond accordingly. Adele will share with you the issue about the presentations. Adele?
Absolutely. Thank you, Yompi. Yes, definitely. And thank you to Dr. Francois and Dr. Ben du Plessis. We really appreciate it. This was the 6th webinar that the DSA had for this year. And for any of you who have missed the previous ones, please go to our website, www.foodfocus.co.za, and then you will see at the top, there's a bar that says resources. And if you hover with your mouse over it, it will bring up all the webinars with the recordings. So for all the people who have asked in terms of the presentation, yes, what we will do is we will edit the recording, and then we will post it onto the website, and we will also have the slides in a PDF that you can then download. I also want you to remember that on the 30th, we've got another webinar scheduled with the DSA, and this one is specifically around key areas of hygienic design. So please make sure that you also do register to attend this webinar. And I think then from my side, I just want to say thank you and wish you all a blessed Friday and a wonderful weekend. And go well. Thank you, Yompi.
Thank you to all. Thank you very much.
Thank you, everyone.